Hi guys, we are back with the role of vitamins as the cofactors. Now we have seen in our earlier lectures that these vitamins, they function as cofactors or coenzymes and act as a transient intermediate carrier of a particular group. And the vitamins which are very famous in this series, they are the various vitamins of the B complex group. In our earlier video, we talked about riboflavin. Let us today talk about another particular vitamin discovered long back in 1938 by Roger Williams from yeast and liver extracts. Though it is found in all plants and in all animal tissues, the vitamin, yes, is vitamin B5, that is your pantothenic acid. Now this vitamin is very different, different in the sense that it acts as a carrier of a particular group. As I told here, acyl group in general is transferred by this particular pantothenic acid. But this, it acts in the bound form. It is available in the bound form in a more elaborative structure of coenzyme A. So if you look at the structure of a typical pantothenic acid, you will find that this is what a pantothenic acid it looks like. Yes, and it has a carboxyl group at one end and it has a hydroxyl group at the other. So this is the structure of your vitamin B5, that is your pantothenic acid. Remember, at one side you have a carboxyl group, at other side you have the hydroxyl group. Now this particular hydroxyl group, if you see, this is here bound to your ADP moiety, 3'5' phosphate diphosphate ADP moiety. This has bound here with the phosphoanhydride bond. So the hydroxyl group is bound here and you have your carboxyl group bound here to beta mercaptoethanolamine. So we have beta mercaptoethanolamine. Yes. You have this particular pantothenic acid in between. So you have this pantothenic acid which is in the bound form in between. You have this 3'5' what you call as ADP moiety here. You have the adenine, as you can see here. You have the ribose 3-phosphate. So this forms the structure of your entire coenzyme A. This was called as coenzyme A because its role was discovered in the process of acetylation. Now today we know that it is not only involved in transfer of acetyl group but is involved in the transfer of acyl groups in general. So this is your entire coenzyme A and this is the reactive thiol group. So this is actually the group where we are going to focus on. So reactive sulfhydryl group or the thiol group, where your acetyl or acyl group is going to be bound with the formation of a thioester. Now generally esters you have heard in the terms of what you call as your oxygen esters. These are different esters. Instead of ROH you have a RSH here. So these esters you will find that they are more what you call as good in transfer of these particular acyl groups. So yes, it serves as a transient intermediate carrier of acetyl group or acyl group. Here you can see this is a structure of a typical acetyl CoA. So acetyl CoA, a coenzyme A carrying this acetyl group. So this acetyl group is bound here transiently. So you have a CoAsh which has that SH group. You transfer the acetyl group here and then this acetyl CoA can now transfer this particular acetyl group to the acceptor. So this acyl group is carried from the donor to the acceptor. So the transient carrier of acyl group. So the vitamin which we talked about, as I told, is your pantothenic acid. Pantothenic acid, remember the number, vitamin B5. The cofactor form is your coenzyme A. Remember the coenzyme A, this entire structure containing this ribose 3-phosphate, this adenine, pantothenic acid and beta mercaptoethanolamine. This entire forms this coenzyme A. The group transferred is acyl group in general, and the enzymes which are being transferred or which are what you call as having these as the cofactors, it includes your pyruvate dehydrogenase complex and your citrate synthase. So we'll have a deeper look into how this group is being transferred. So let us look at these particular two enzymes, that is your pyruvate dehydrogenase complex and citrate synthase. which have this coenzyme A as its cofactor for the transient intermediate transfer of this particular acetyl group. So we know that this pyruvate dehydrogenase complex, it catalyzes the oxidative decarboxylation of pyruvate. So pyruvate, it is what you call as oxidized, it is decarboxylated, and this particular acetyl group is transferred to what you call as CoA to form acetyl-CoA. Remember the thiol group which we saw, and that particular thiol group, it forms a ester with this, and this forms what is called as your acetyl-CoA. Now this acetyl group transiently attached to CoA will be now transferred to oxaloacetate. The first step of your what you call as linking reaction or formation of what is called as your citrate in your tricarboxylic acid cycle or the Krebs cycle. So this particular citrate synthase now will take this particular acetyl group from acetyl-CoA, attach it to this oxaloacetate and form what is called as your citrate, and your CoAsh is released, which can now again act as a carrier of the next acetyl group. So this is how your vitamin B5 or pantothenic acid, it works in the form of coenzyme A for the transient intermediate carrying of acetyl groups and acyl groups in general. So stay tuned with us, with me, Professor Girish Kukreja, for more in microbiology, biochemistry and much more. Thank you.